Our first speaker is JB Lalanne, who's a new PI at UDM in Montreal. He's got a really kind of unique combination of expertise in experimental and theoretical sciences, and been studying regulation across many model organisms in bacteria, now mammalian systems. And he's had a number of amazing papers, too many to highlight here, but just to say, you can go look at his publication list to learn more about this work he has done before. And we're very excited to have JD speaking today. So it's over to you.
All right, great. Perfect. Thank you so much, Ben. That's really kind. And whoops, sorry. I guess I'll jump. My title slide got messed up. Sorry about that. So I'll jump right in. My name is JB Lalanne, as Ben mentioned. I'm at UDM in the Department of Biochemistry, and the fragile nucleosome is like pretty much one of my favorite virtual seminars. So it's a real privilege to be here. And so, as Ben mentioned, like many people in the audience here today, what the focus of my lab is studying how the genome quantitatively encodes regulatory information. And I like to start with this sort of early mouse development picture where we see that differential expression of transcription factors, here GATA4 from the endoderm lineage and NANOG from the sort of pluripotent lineage, really is what drives these early cell fate transition. And when we look under the hood of this process in the neighborhood, say, of this transcription factor GATA4, we see a very dynamic chromatin landscape, right? So what you're seeing here is chromatin accessibility in both pluripotent and endoderm lineages. And you can appreciate that there are many regions that are completely differentially accessible, right? So, the canonical developmental enhancer is going to be closed in a certain cell state. And as the cell fate transitions, these regions of chromatin open up as a result of differential expression of transcription factors that bind and evict the nucleosome and eventually regulate the expression of genes. So what my lab studies is really the sort of sequence to function relationship of these regulatory elements. And so if we zoom in the genome to these regions, where we can see the sort of sequence of DNA, we can also map the binding sites of important transcription factors that regulate the process. And so the whole question that we're studying is how the arrangement, affinity, number, position, orientation of these transcription factor binding sites effectively leads to quantitative regulatory output. Okay, and so this is the so-called cis-regulatory grammar. And of course, if we're trying to understand gene regulation, it's not just the process that's happening at a single enhancer, it's at the whole locus. There are many enhancers that are contributing to gene regulation, but I would argue that even the single enhancer process is not completely resolved. So what my lab really focuses on is the sequence to function relationship at the level of individual enhancers. And so how do we study this process? A sort of remarkable property of regulatory DNA is that they're sufficient to produce really interesting biological activity. And so the sort of empirical phenomenon is that we can take these sequences of DNA and clone them in transgenes, in reporter transgenes, where we effectively just create plasmids where we take the CREs, the cis-regulatory elements or enhancers, clone them upstream of, for instance, a fluorescence protein. We can introduce that construct in our system of interest. We work a lot with cell lines and stem cells. And then we can differentiate these cells into different kinds of organoids. And what's really remarkable is that these regulatory fragments effectively lead to very stereotypical cell type specific expression in different systems. And here I'm showing our stem cell sort of embryonic body model, but it's a very generic phenomenon that's seen across metazoa. Okay, and so that allows us to study these regulatory elements at a relatively high scale, but these reporter assays, as I'm showing here, historically were done in a sort of one at a time manner. And what my lab has been working on and developing is sort of how to achieve much higher scaled measurements to study these sequence to function relationships in regulatory elements. And so I'll just briefly start by introducing the concept of barcoding and massively parallel reporter assays that many might already be familiar with, but just to make sure we're all on the same page. And so, as I alluded to, the historical way that people were studying regulatory elements was to just take these cis-regulatory elements, clone them in reporters, and as a result of the biological activity of these elements, these would produce mRNA molecules that could be measured upon transfer in the biological system of interest. This sort of really key innovation that happened over 10 years ago now was that if we could combine those with random DNA barcoding, which are effectively tags that can be added to the construct, and if these random tags can be matched to enhancers, that really allows us to make highly scaled measurement of enhancer activity. And so the whole pipeline is effectively that we can clone super complex libraries of either enhancer variants or different enhancers, introduce those into cells, extract the RNA and the DNA, and then quantify activity using next generation sequencing. And so this, again, is a relatively old pipeline, but it's extremely powerful, and it's been used very widely to study gene regulation and the cis-regulatory grammar. And so one of the limitation of this, these so-called massively parallel reporter assays is that historically they've been mostly used in static contexts. And by that, I mean sort of workhorse cell lines, such as the ENCODE cell lines. And again, I don't want to minimize the power of the methods because they allow us to study, upwards of hundreds of thousands of regulatory elements in a single experiment. So they're extremely powerful, but a lot of interesting biology actually happens in dynamic heterogeneous system that change over time, that are multicellular. And so I mentioned organoids earlier, but we can think about in vivo systems like the brain, which has hundreds, if not thousands, of different cell types, or disease modeling, where there's progression of cell fates within the system. And so these assays, as they were developed historically, didn't have the power to resolve activity over the different cell types. And so one of my key expertise has been to effectively develop the next generation of multiplexed reporter assays that combine the power of barcoding to generate highly scaled enhancer activity with the sort of ever-evolving suite of single-cell RNA-seq technologies. And so without going into the details, single-cell RNA-seq allows us to generate a complex census of the different cell types and cell states in complex biological systems. And so it's a very natural desire to combine these barcoded assays with the single cell technologies. And so because this is published, I won't go too deep in the details today, but we've been establishing these single cell quantitative expression reporters. We're calling them skewers as a sort of playful analogy because they're multiple. These new classes of reporter combine different barcoded RNA molecules, but the upshot is that this new barcoding technology allows us to generate single cell maps of enhancer activity at very high resolution, very high signal to noise, and importantly, in a fully multiplexed manner. And so we're very excited by the technology because it allows us to sort of measure at single cell resolution the activity of enhancers and more specifically, their quantitative cell type specific activity. And so what I'm showing here is just an example of a pilot experiment that we did to profile the activity of hundreds of putative regulatory elements in our organoid system. And again, these are all through barcoding, but you can think of these maps as sort of these in situ assays, but instead of being done in a one at a time manner, they're done in a sort of completely parallelized fashion. And so we have this technology that allows us to screen for regulatory elements that have really interesting biological activity. And for the sort of rest of the talk, I'll be talking about vignettes on dissecting the sequence of function relationships of some of these developmental enhancers. And so what my lab focuses on, again, is leveraging these barcoding technologies to study how enhancers work. So today I'll be talking about examples of the kinds of assays that we can do to really start dissecting very quantitatively the sequence function relationships of enhancers. We also try to address the question how to make better enhancers. And by that, I mean stronger, more active, more compact elements. And so we use both model-driven mutagenesis using sort of deep learning strategies. I'll also be talking about our thread on sequence transforming enhancers or torturing the sequence, so to speak, of regulatory elements to generate variants that might have higher activity. And we also study how enhancers evolve by testing the activity of orthologous sequences sampled from the mammalian phylogeny. But I won't have time to talk about that today. So let me sort of, again, reiterate the key question that that my lab focuses on, which is this sequence to function relationship between the DNA sequence of an enhancer and the biological function, which is cell type specific expression. And what's fascinating to me is that even though it's an extremely well-studied area, there's still simple questions that don't have sort of definite answers, that we don't have definite answers to. So one simple question that we could frame is, what is the minimal size of an enhancer? Here I'm showing about a kilobase of DNA, and we know that a few hundred base pairs is enough, but can we quantitatively delineate that? How robust are enhancers to mutation, right? Like, are certain enhancers more fragile or other enhancers more robust? How can we assess that? And more broadly, what classes of sequence perturbations are enabling to retain regulatory function? And so I'll touch on these three sort of questions sequentially in the rest of today's presentation. So as in every other investigation, we need a model system. And the model systems that we've been focusing on are these endoderm-specific enhancers. And I sort of started the talk talking about this GATA4 enhancer. These are just the single cell activity maps from the barcoded assay that I've shown. And as you can appreciate, all these 5 enhancers have very sort of specific endoderm lineage activity. And so we see them as a nice sort of model of these class of regulatory elements. And the other thing that is particularly interesting to us is that the set of transcription factors that are known to regulate gene expression in the endoderm lineage are relatively well known. Here I'm showing a few examples: GATA4, GATA6, KLF4, SOX17, FOXA2. We can map those transcription factor binding sites on those model enhancers, and they all have a very high density of these putatively implicated endoderm lineage transcription factors. And so we have a very strong molecular prior as to what are the important features within those enhancers, and that allows us to more easily interpret our result. And so using these model sequences as a sort of test ground, we've been generating different classes of variants to test the activity, right? So I'll be talking about experiments where we basically test the activity of subtiles within those enhancers. And even though subtiling has been done many times in the literature, no one's really looked at the size, the importance of sort of testing subtiles of different sizes, right? So I'll be talking about that. We also do saturation mutagenesis, where we perturb every base within these enhancers and test the impact for function. And again, we do funkier things where we torture the sequence of these enhancers and measure the impact on function. And we also do other things such as optimization and compaction using models, but I won't have time to talk about that today. So just to sort of finally dive in the kind of data that we can generate on this subtile scanning. And to orient you, what we can do is we can take a kilobase of DNA that again corresponds to these developmental enhancers, and we can synthesize short subtiles that we just kind of scan along the regulatory element. And using barcoded assays, we can measure the activity of these subtiles. And so that's the kind of data that we can generate, this MPRA activity, and you might have trouble sort of seeing the gray line here, but for very short tiles of 40 base pairs, perhaps unsurprisingly, we don't see any activity over our minimal promoter only background, right? And we can sort of mark the maximum activity as a function of tile size. But as I mentioned, what's really cool is that we can do this same process, but now with tiles of progressively larger and larger sizes. So you can see that at the level of 70 base pairs, now we cover sort of regions that include more transcription factor binding sites that I'm sort of schematically representing here. Um, and we now start seeing activity over background. And we can just kind of do this process with larger and larger subtiles, always scanning those tiles along the sequence. And we do that, we sort of start seeing a sort of very nice, highly nonlinear increase in activity as we increase the regulatory size. And so other things that are nice to note is that in black here, what I'm showing is the accessibility. So somewhat unsurprisingly, there's a very nice correspondence between the activity of the tiles as measured by these reporter assays and the accessibility, right? Again, not extremely surprising, but very nice to see a sort of concordance there. But what I want to draw your attention to is this highly nonlinear increase in activity, okay? And we can repeat this process across our different model enhancers, and we see, even though it's really fascinating because each enhancer is sort of its own universe in a way, and even though they behave quantitatively differently, we see emergence of similar patterns, such as this nonlinear activation. This is a log scale, right? So in all cases, we see this highly synergistic effect where progressively larger tiles really start boosting activity in a nonlinear manner. And so there's a lot of quantitative information in these data that I don't have time to talk about, but that provide hints about mechanisms in the activity of regulatory elements. But because this is a full multi-scale assessment, the next thing we did was to basically take high activity tiles and really dissect at high resolution. So meaning generate these saturation mutagenesis maps of activity where we go in and generate all possible single nucleotide variants and measure their activity again using our barcoded assays. And so the kind of map that we can generate in that way looks like this. So this is to orient you a 300 base pair high activity tile for one of the enhancers that we've been studying. And we can, again, for every mutation within that enhancer, measure the activity. And if you focus on this representation here at the top, pockets of blue corresponds to regions where mutations are deleterious for activity. And so if you know about gene regulation, you're probably guessing that these regions correspond to binding site for transcription factors, and that's indeed the case, right? What we can do is, because we have quantitative models for the biochemical affinity of these endoderm lineage transcription factor, we can map those along our sequences, and we actually see a very beautiful concordance between pockets of activity, pockets of regions that are important for activity, and mappable binding sites to these endoderm specific lineages. So doing that, we can start building very quantitative maps of enhancer function, not only at the level of the full kilobase, but down to the sort of single base pair resolution. And we can even interrelate these two classes of data, the subtiling and the saturation mutagenesis. So for instance, I mentioned this sort of synergy between tiles. If we look at two short subtiles that cover only a subset of the transcription factor binding site within the enhancer, the activity of these short subtiles is very low, very close to the sort of baseline in our assay. But when we consider the full tile that covers these set of 7 transcription factor binding sites, then we get sort of full activity compared to wild type. So that's the kind of synergistic interactions that I was talking about that we're trying to sort of dissect in more molecular details. But the sort of entry point are these quantitative measurements at the level of the sort of using our barcoded assays. And so what's really interesting is that we can generate these maps of these sort of high resolution map using saturation mutagenesis across different enhancers. And we see very interesting phenomena such as differential robustness, as I alluded to in the beginning. Some enhancers are highly fragile, such as this one, many, many mutations, or break it, whereas others are much more robust, and we're also exploring the sort of determinants of that. But what it's also highlighting is that there are very stiff directions in regulatory sequence space whereby a single base pair mutation in a key transcription factor can completely sort of abrogate activity, which is perhaps not too surprising, but which is kind of speaks to the sort of fragility of some of these elements. But I'll just end with this final section of trying to explore larger scale perturbations to enhancers that are much sort of a much larger disruption of the sequence space, but that can lead to retained function. Okay. And that gets at this sort of very I'm sure everybody in the audience is sort of familiar with these canonical ideas, but this idea of the robustness of the cis regulatory grammar, meaning how flexible is the arrangement of transcription factor binding sites in enhancers. And there are these two very sort of well-known canonical extremal expectations, one being that transcription factor binding sites are completely flexible on sort of DNA, and maybe moving them around in the so-called billboard view doesn't affect function. And the other extreme is a highly rigid view where any deviation leads to loss of function. And there's evidence pointing to both, and obviously it's sort of more complicated than these two extremes, but they provide a useful sort of conceptual framework to think about the problem. So what we've been exploring, which is novel, is these sequence transformations of enhancers. And we've called that enhancer derivatization. And the idea is that you can start with a wild type sequence that has function, as I've shown, and you can now anchor your view on these key transcription factor binding sites, and you can generate sort of enhancer derivatives through just permutation of the positions, and as I'll discuss a few different classes of sequence transformations, and all these enhancers sort of look like the wild type sequences, but they're derived from that wild type sequences. And I like to sort of show this analogy of the derivative product, right? In the context of enhancers, we know that the starting point has function, but what about these sequence transformed elements? Do they all have function? Is it a complex? Is it more complex, right? So these are the kinds of questions that we've been exploring. But what's really nice is that because these are statistical processes, we can effectively generate as many of these enhancer derivatives as we want and test them experimentally. And so we can really get at this question of robustness in a highly statistical manner. And so the kind of sequence transformations that we've been exploring is taking our transcription factor binding sites from our developmental enhancers and depositing them in the fixed position and orientation across random DNA background. We've also taken our binding sites and deposited them randomly, again, in background DNA. We've also done things where we cut our enhancer in pieces and reassemble, right? And we can generate data for all these sequence transformations. And more concretely, what that looks like is something like this, where we just, in the case of what I've called synthetic trypsis, right? Breaking the enhancer and putting it back together, it sort of looks like this, right? And we can repeat this process over and over again. So concretely, if we have an enhancer of interest, we can randomly pick breakpoints, reassemble the sequences, and so that's sort of what the reassembled enhancers would look like. This is where the binding sites are if we map them. But importantly, we can measure the activity of all these sort of reassembled enhancers. And so what you're seeing here is the baseline minimal promoter activity. This is the activity of the wild type sequence. And really remarkably, these sort of fully reshuffled enhancers, in many cases, retain with an experimental error the activity of wild type. The vast majority of them are still active, and there are even some that are more active than wild type, right? So we see that as a really interesting phenomenon. So I'm out of time here, but I'll just end by saying that we can use um, these sequenced transformations to generate many sort of different measurements of enhancer activity in a way that is really interesting because we can completely sort of mess up the sequence of the enhancers and we retain activity as opposed to sort of single base pair mutation that can completely abrogate it, right? So that's what I'm calling the soft and stiff directions of regulatory sequence space, and that's what we're exploring using these different variants. So with that, I'll just end by saying that I'll just end by acknowledging the people in my lab, my funding sources, and sorry, that was a little quick, but thank you so much for your attention. I'm happy to take questions if there are any.
Awesome. That was a fantastic talk. We have a couple minutes for questions, and maybe I'll start.
Yeah,
So with that bit at the end where you're shuffling the positions
yeah,
and you're seeing like restoration, and it says an endoderm enhancer, you're seeing restoration of activity.
yeah,
Have you looked to see if you lose specificity?
yeah.
Like
That's
do you
a
see, yeah.
Yeah, that's a really fantastic question, especially for the ones that are, more active than wild type. We're really interested in the question of trade-off between activity and specificity. And so we haven't, this was all done in a sort of endoderm cell line. So we haven't yet tested these in our multicellular model, but that's definitely something we want to explore. There's no, you could imagine sort of having an optimized grammar where, and people have reported that, notably Emma Farley in other systems, where, there's definitely a trade-off between activity and specificity. Unclear here if that applies, but that's a really good question. Uh,
Awesome. Anyone in the audience has more questions, you can type those into the Q&A. And JB, you can answer their questions there by chat. I'm
Great.
just going to post to everyone the sign up sheet if you'd like to be considered for a talk in the new year.